And now, to conclude the morning session, it gives me great pleasure to introduce Professor Ueli Rutishauser. He's coming to us from the Cedars-Sinai Medical Center in Los Angeles, where he is assistant professor and director of the Human Neurophysiology Research Laboratory. Professor Rutishauser has a, so far, brief but brilliant career that happens to have elements that resonate almost perfectly with just about everything we are aspiring to here at the Allen Institute. In particular, to build those bridges from the activity of individual neurons in the brain to the activity of networks of neurons in the brain, and from there to phenomena of behavior, perception, and indeed understanding, and in this case, truly bridging into the realm of human cognition. His background and training also bridges realms from the cell biology of neurons. He trained at Caltech with one person who is one of the world's most esteemed neuronal cell biologists, Aaron Schumann. And also, indicative of the breadth of his interests, was jointly supervised in his doctoral research by a chief scientific officer of the Allen Institute that many of us know fairly well, Christoph Koch. But you know, that's, from the very beginning, this guy has been sort of like a one-man Allen Institute in microcosm. So I think he is, you know, a particularly wonderful choice to be the day's first of several wonderful Allen showcase next generation leader speakers. And I think probably about now you'd rather hear from him than more of my perambulations. So, Professor Rutishauser.
Thank you for that very kind and generous introduction, and welcome everybody. It's a great honor for me to be here and to speak to you today about my research concerning recordings in humans, very much like what we just heard about in animals, but we try to do some of these experiments in awake behaving humans. And as you will see, this comes with its unique set of problems, obviously, but to some degree we can do some of the things that we just heard about this morning. So when we think about, and when we want to study human cognition at the single unit level, that seems at first sight something which is entirely impossible and inconceivable. However, there are, as a matter of fact, a number of opportunities in clinical neuroscience, in neurosurgery, which permit these type of experiments to a very limited degree. Now, there are a surprising number of these opportunities, and as you will see, they provide exceptionally valuable data, which otherwise is not obtainable in any way. And apart from being useful to learn about how the brain works and how the brain learns and how it provides cognition, these experiments also are really motivating the development of animal models. And the thought is that a few key experiments which replicate phenomena which can be seen in animal experiments as well as in human imaging would be able to bridge these two worlds and suddenly make a particular experiment very relevant, for example, for autism, as you will see. There are two clinical situations among the few that exist that I'm utilizing for my research are the following two. One of which is the intraoperative recordings, which is number one here. These recordings are done while electrodes are allowed into the brain to to locate the appropriate target for the placement of a deep brain stimulation electrode. Deep brain stimulation electrodes are only effective if they're located in the appropriate target structure. And it turns out that the best way to locate that structure is to do microelectrode recordings. And by carefully watching the waveforms that one sees during this operation, one can find the appropriate target structure. But apart from that, this also requires that the patient is awake. Thus, we can do experiments while we do this. And here I'm showing you an example that we did in my lab from the substantia nigra. The second situation, certainly much better known, is epileptic seizure localization. It turns out that in many cases where the seizures are in principle surgically treatable, it's not known where the seizures are coming from. This requires implantation of electrodes in various locations, and then these electrodes are chronically implanted. The patient goes back to the unit in the hospital and waits there till the seizure occurs, and this will then allow us to localize the seizure. Here I'm showing 3 examples of the kind of recordings that this permits. Now, focusing on the epilepsy situation, this is the type of electrodes that we use. Now, clinicians are interested in the signal that's provided by this electrode. This is called a macroelectrode. It has these big metal contacts, as you can see. And these provide what's called an intracranial EEG across the depth of the cortex all the way to the midline. This would be lateral, this would be medial. At the tip of this electrode, this is a zoom-in, there are additional microwires which extend like this here. These are tiny microwires, 40 microns in diameter, and these wires are inserted for research purposes only, and they permit recording single neurons. And safety studies have been conducted, and in fact, this is an FDA-approved electrode that one can use. And all the data that I will be showing in the following slides originates from the tip of these type of wires. After implantation, the patients undergo an MRI, a structural MRI, and this is an example of one of my patients where you can see the electrodes going in from lateral to medial, and I have marked here as an example with the arrowheads the amygdala and the hippocampus sites, which with the tip of the microwire is indicated by the arrowhead. These surgeries are fairly stereotypical, meaning one can repeatedly target the same brain area reliably in a large group of patients. And this is, for example, illustrated here, where we have marked with a colored dot the tip of the microwires in a group of patients where we aim to record from a certain part of the amygdala to compare epilepsy patients with and without autism to see whether we could attribute specific phenomena to this particular brain area. Now, today I will focus on a study that concerns learning. In particular, this study that I will show you data about concerns the single neuron mechanisms of single trial learning, such as when you this morning came in here and met a person you have never seen before. The question is, what happens in the brain? And how come next time you see this person, 10 years later, you most likely will still have a very strong feeling of familiarity? For a lack of time, I won't talk about these other experiments that I have been working on, but I'm just listing them in case you're interested, and feel free to ask me about them later. So my strategy to study memory formation is the following. This is a very simple, but as you will see, very powerful experimental strategy to test memory formation and to test decision-making about internal states. What I'm doing is I'm showing patients images, such as this woman, which simply you have never seen before. You look at this image for a second. The neuron might not care about this very much. I'm showing you some other images in between, and then I'm showing you this same image again. Now the question is, have you seen this image before? Yes, you have. And well, when you saw it before, what was the color of the dot? Well, it was green, so that's an associative memory. This very simple experimental paradigm is very powerful, partially for the reason that it requires a very rapid formal learning. You might have seen this picture only for 1 second, and you have created a long-lasting memory. So this requires very rapid plasticity. Now, this is the task that we have parameterized this behavior. So if you're a patient doing this experiment, you will be seeing a sequence of images on the screen. Each square here indicates the screen. You look at this image for a while, for a second here, and then you're asked, have you seen this image before? And you have to decide an answer on this scale, new or old, and how confident are you? Now, some of these images you have never seen before, and some of them you have seen once before for 1 second. And the type of neuronal response that one sees is exemplified well by this individual neuron. So what you're seeing on this slide is one single neuron in the hippocampus of a patient. And I'd like to point out a few key features of this neuron. So here the green trials are learning trials. The patient looked at a variety of different images, a tree, a car, a house, and the neuron didn't change its firing rate. Its mean firing rate remained the same. However, during the later memory test, the very same images were shown again on the screen. These are the red trials. And now the neuron responds very strongly. So this is a neuron which was clearly not responding to the sensory stimulus alone because the sensory input was the same. In contrast, this neuron, in fact, represents the internal brain state that this person has already seen these stimuli before. Now, what are these neurons and what are they doing? Do they have anything to do with memory? And I will show you now a series of experiments to convince you that, in fact, these neurons are a carrier of memory strength. Now, when we think about memory strength, what does that mean? Well, so memory strength can be weak or strong, in which case you will be answering this question accurately or inaccurately. And so this means there must be a representation of uncertainty in the brain. And there have been numerous hypotheses and theories on how uncertainty would be represented, but from the point of view of signal detection theory, this would be the most natural way to think about it, that there is a decision variable in the brain which value scales in some continuous fashion as a function of memory strength. And then you would have different decision thresholds to answer the first question, have you seen it, yes or no? And how sure are you? You perhaps place the threshold here. Now, from the point of view of neural computation, it has increasingly become clear that a model like this is at work, because if one considers what is the optimal way to compute from a Bayesian point of view, for example, it is very clear that a representation of uncertainty, a representation of variance, is an integral part of any computation at all. This is very much in contrast to what classically in psychology people have described, where the judgment of confidence or signal reliability is ascribed to processes often labeled as metacognitive, which are supposed to be some sort of separate process, which happens in these models often after a decision has been made. And I will show you that, in fact, the human data is very much compatible with the first signal theoretic interpretation. Now, I'll first show you some behavior to show you that patients are indeed very well able to discriminate between weak and strong memories. So, for example, here I'm showing you the behavioral performance when patients indicated that they were high, or they had high or low confidence. And as you can see, most of them were clearly able to differentiate between high and low confidence memories. Now, this is further substantiated by decision time data, which is reaction time data. In memory experiments, there are certain signatures of decision times which one sees over and over again, which allow one to conclude that a memory is declarative and thus hippocampal based. And these signatures are the following, which naively seen seem actually rather counterintuitive, but you will see that this is really what one would expect from this kind of data. So most importantly, the finding is that high confidence decisions are made faster than low confidence decisions. And we can see this here. New decisions take more time than old decisions. That's also important, but a repeated finding. And then it's also the case that correct decisions are both made faster as well as with higher confidence. And the patients very much showed these patterns. Thus, we can be confident that this behavior is a true declarative memory. Now, going to the electrophysiology, the type of data that I'm going to describe is based on these type of recordings. I'm showing you here an example recordings from the hippocampus of a patient. This is the broadband signal. This is the filtered signal. These are some isolated neurons from this particular recording site. And the data set that I'm going to describe consists of 1,065 neurons pulled from the hippocampus and the amygdala. These neurons are surprisingly visually responsive. So a very simple comparison, baseline versus stimulus, usually yields about 30% of neurons are responsive in this very high-level brain area. Which is really surprising. So first of all, I'm going to describe one population of neurons, which I'm calling NO neurons, newlold differentiating neurons. These neurons are identified by comparing the mean firing rate in a large window, 1.5 seconds, after stimulus onset between new and old stimuli, only considering correct trials. The patients always answered correctly. And there's two types of these neurons that one can find at first sight. One of which is this one, which is it increases its firing rate maximally for new stimuli. These are true negatives. The patient correctly found these as new. Whereas the other group of neurons does the same, but it increases its firing rate for old stimuli. And in this particular data set, there were 81 of these neurons. Now, when you look at these rasters here, I have sorted them according to behavioral confidence that the patient used. And you can appreciate that both of these rasters show a gradient in that firing rate here is maximal for high confidence new decisions, and firing rate here is maximal for high confidence old decisions. So there seems to be a gradient of signal strength that these neurons have according to behavioral confidence, which is a behavioral readout of these neurons. Now, quantifying this using a single neuron ROC analysis, I find that this is indeed the case. So for the two same example neurons, I'm showing you here a single neuron ROC computed from both low and high confidence trials separately. And it's apparent that there is more signal on a single trial basis for high confidence compared to low confidence trials. Now, it's important to note that in both cases, the answer that the patient gave was the same. It's irrelevant. I'm just asking, is it new or old? So the decision in either case was correct, but there's still a difference in signal. And it turns out that if one does a population level analysis, that this is indeed a reliable effect. So what I'm showing you here is a histogram of the area under the curve of these single neuron ROCs for all the novelty sensitive neurons, for high confidence and low confidence neurons here in this mirrored representation, and here in a paired representation. And indeed, this was the case for both subtypes of neurons. And interestingly, this is even the case if one compares the ROC curves of low confidence trials and false negative trials, which is not shown on this slide. So false negative trials are trials you forgot. You said, oh, they're new, but in fact they were old. And one could consider those as some sort of a very low confidence trials, but the neurons still had a signal in these cases, meaning, and I have shown this previously, that when one trains a decoder, one can in fact outperform the patient quite considerably this way by using this signal. Now, these two types of neurons at first sight seem to be the same, but just coding signals with opposite sign. However, this is not the case when one looks at this more closely, and I will show you that this, from a decision theoretic point of view, actually makes a lot of sense. So from the example of old, bigger, new neurons, I'm showing you here the distribution of firing rates using a CDF for old, high confidence, old, low confidence trials. You can see clearly the two distributions are different, versus for new trials, low versus high, the distributions were not different, and this is summarized here. However, this neuron still does give you information about new trials because it reduces its firing rate below baseline in new trials, but it does not differentiate confidence. And the opposite is true for new, bigger, old neurons. So if one would only like to know whether a stimulus is new or old, listening to either group of neurons would be sufficient. But if one would like to also distinguish between high and low confidence trials or strong and weak memories, one would have to listen to the appropriate subset of neurons. And without going into detail, it in fact turns out that this is the optimal way to code this information if one assumes that the readout is a mechanism which is based on the difference in firing rates between these two neurons, which is this here. So if one considers a decision-making model which accumulates or integrates the difference between these two neurons, this would indeed be the optimal way to actually do it. Now, let me turn your attention to a different group of neurons, and in a few slides it will become apparent how these are relevant to the discussion that I just did about the novelty neurons. These neurons are what I term visual category neurons. These are visually responsive neurons, and their response indicates visual properties of what's on the screen. In particular, it indicates what's the category of the stimulus. For example, listening to these neurons would allow you to tell whether there is an animal or a car on the screen. So for example, this particular neuron here on the left, it only increased its firing rate, as you can see here, the red trials, if a fruit was shown on the screen, but not for these other type of images. Similarly, this neuron increased its firing rate to foods, whereas there's another group of neurons which has a much more complex mixed type coding, but nevertheless is strongly modulated by these abstract attributes of the stimulus. Now, these neurons are fairly common. They're about up to 20%, in this case 17.5% of the neurons were of this kind, and they are largely distinct from novelty neurons. Now, these neurons, interestingly, are completely insensitive to memory. So if one repeats a similar single neuron ROC analysis to quantify how well these neurons distinguish what is shown on the screen, that is not sensitive to whether the patient retrieved this memory with low or high confidence, whether the stimulus was new or old, and other attributes that are not shown on here, and the distributions are completely overlapping. Thus, these neurons seem to really be input-driven and sensory. They are not sensitive to memory retrieval. Now, this is interesting if one considers the latency of these neurons. So what I'm showing on this slide is a differential latency, which means what is the first point of time if you listen to an individual neuron that you could distinguish between its preferred and non-preferred stimulus. And note that this concept is different from an onset latency. It's a differential latency. If one uses a concept of differential latency, which relies on the cumulative firing rate of all the neurons, one finds that on average, these category neurons distinguished between the categories after 272 milliseconds. That's this arrow. Whereas in contrast, the novelty neurons took up to 180 milliseconds longer to make this decision. So on average, 461 milliseconds. And note that both neurons were selected using the same time window, so that can't explain this difference. So the question is, what happens during this 180 milliseconds, which, if one considers synaptic delays, spike timing, that's a really long time. And during this period, we believe this is the appropriate period to study memory formation, as I will show you. We next considered this process from the population level. So I've only analyzed these selected groups of neurons. Of course, the majority of neurons was not considered for that analysis, so it could be that I'm missing the crucial aspect, the interaction between these two neurons. So to address this question more generally, we next produced a generalized linear model of the spiking response. And this model is used to explain the firing rate as a function of time, as a function of the stimulus properties, which are, is the stimulus new or old, which category is it, and is there an interaction? And quantifying the percentage variance explained using omega square of these different variables, one sees indeed that there are two waves of information that are going through this population. And the first wave is visual. It tells here in this case what is the visual category of the stimulus. Then the second wave is memory driven. It's new versus old. And that second wave is the most surprising to me, because remember, these patients, what differentiates new versus old is a single 1 second presentation. So it represents a very rapid change in this population. And using this this regression model of the single neurons, we can now indeed see that these two populations code information orthogonally. So if one considers the omega square only for the novelty neurons, one can see that they contain no visual information. They contain only new old information, and vice versa here. And again, that only these NO neurons are sensitive to memory strength, as indicated by confidence, and they're not sensitive to, the category neurons are not sensitive to this. And indeed, if one does a correlation between the different effect sizes, one finds that the average correlation between these effect sizes is 0, meaning it's an orthogonal code. These two neurons have no interaction, and the interaction terms are insignificant. And there are clearly a few notable exceptions. For example, these two, they are strongly significant for both types. But these are very rare, and we are still trying to understand what this means. So for example, this could be a neuron which says novel animal. But those seem to be very rare and on average actually not significant. So this regression model is very useful to quantify the pieces of information and the time course of information. However, the values given by these models, 5% variance explained, are not inherently meaningful about neuronal firing rate. What does it mean to explain 5% variance? Well, that's not a very useful number. So to get a more quantitative understanding of what it means to have lower high confidence memories, we constructed a population decoder. And this population decoder allows us to quantify information present in the neuronal population in units of mutual information per time. And I'm plotting here this information in terms of bits per 500 milliseconds. So that's a rate of information. And that's a meaningful number. I'm telling you, well, there's 0.5 bits in this population. That's a useful number to consider. And doing this analysis, and without going into details, it turns out that in fact the difference between new and the high confidence memory is surprisingly large. It turns out that it requires about 3 times as much information to go from a low to a high confidence memory in terms of coding accuracy of spike trains in the brain. To me, this was a surprisingly large number. Now I next try to relate these neuronal subtypes to anatomy in an attempt to look at whether these functional types could possibly be related to different anatomical types. And there are indeed hints that this is the case. And so what one typically sees when one records human single neurons is waveforms like this. So these are waveforms from about 1,000 putative single isolated neurons. And there are different ways to quantify this, but the metric I'm using here is trough to peak time, which is prominently bimodal, and which clusters in a plot like this nicely into 2 clusters, which firing rate does not. And note here the firing rates. These are very representative of the human mean firing rates. They are very low. And there are some higher ups, but they are very rare. Now, interestingly, if one plots this representation of froth to peak time only for these NO neurons, it turns out most of these have short waveforms, and some example of which is shown here. Whereas these category neurons, they are mixed, they're of both types, and this is an example of a long waveform. And statistically, it turns out that over 70% of these NO neurons are short waveforms, whereas the category neurons are not significantly differently distributed. So this indicates that there's potentially a different anatomical subtype of these novelty neurons, which goes along with this functional distinction. Now, unfortunately, in the human brain, what it means to have a short or a long waveform is really unknown. Of course, I could speculate according to inhibitory neurons and pyramidal neurons and so on, but that's really actually unknown. And interestingly, from all the other metrics that I have looked at, it very much looks like to me that these are both pyramidal neurons, except for that they have short waveforms. But that's really unknown. All I have shown so far was relative to stimulus onset. But of course, when I'm showing a picture for 1 or 2 seconds to a human, that human is vigorously exploring that image by actively moving around his or her eyes on the screen. So the question is, well, so these long latencies that I have described, is that really because I'm not aligning to gaze position? And this is one thing which we explored. For example, here you're seeing an image that a patient was viewing on the screen with the instruction of viewing these images carefully for a later memory test. And as you can see, these patients vigorously explored these stimuli and they fixated only on those stimuli and not anywhere else. Some of these they had seen before, some of them they have not, and there are different categories as you notice. So interestingly, we found here that these neurons, which are very high up the visual stream and which are thought to have very large receptive field sizes, are in fact very sensitive to gaze position. So for example, here on the left is an individual neuron which increased its firing rate whenever this patient looked at a human. Now, here 0 is not stimulus onset, but that's fixation onset. So this was highly sensitive to where the patient actually looked. And there were similar neurons which were selective to different categories as well as novelty. So I'm showing here another example. This particular neuron would only increase its firing rate if the patient looked at a face, regardless of whether it was a human or a monkey, just a face. So now what is notable here is that the onset latencies relative to fixation onset are markedly reduced to what I have shown before. So the average onset latency of MTL neurons without taking into account eye movements reported in the literature is 390 milliseconds, whereas the differential latency that we have seen is in fact shorter. And here it looks like that it might be even shorter. So we are trying to explore what this means, and we are trying to explore whether it's even, that is really indeed the case that these receptive fields in these high-level structures in the medial temporal lobe in humans are indeed that large, which is a fact we infer from the primate literature. So to conclude the first part of the talk on functional cell types, I have shown you that there are CAT and NO neurons, the abbreviations that I used, which are functionally distinct, and they are functional subtypes. And they are different in the following features. These CAT neurons, they were visually tuned, visually responsive. They respond early, and they're not sensitive to memory. In contrast, these NO neurons are not visually tuned. They are highly sensitive to memory strength. They correlate with behavioral report, and they respond considerably later. The extracellular waveforms suggest indeed that there is potentially 2 different anatomical subtypes which one could associate with these functional types. And that's a question that I would like to investigate further. So these 2 types of neurons, if you think about memory formation and the time delay between them, we think are a potential substrate for memory formation. Because if you like to form a memory, the classical model would assume that first you would like to have very high-level, high-dimensional features, such as is it an animal, is it a cat, is it a tree, and you could then use that feature vector to encode a new memory. And thus the interaction between these two neurons are a potential substrate for memory formation. And the NO neurons, there were two subtypes, these N bigger O and O bigger N neurons, which at first has always puzzled me, but after theoretical considerations of integration models, it finally became clear to me that this is actually an optimal way to code this information if you believe that this decision is being made, perhaps in LIP, by an integrator which reads off these neurons. So before concluding, I would like to make a few general observations about the approach that I have taken and the approach that this offers. Now, these neurosurgical procedures provide exceptionally valuable data, and keeping this in mind, of course, these type of experiments are necessarily incredibly limited in that I cannot do many of the anatomical procedures that I would dream of doing or like of doing. But despite all these constraints, these experiments have in fact provided the most direct link we have at present between individual neuronal activity and actual memory, which means behaviorally measurable declarative memory that I can ask a person to declare verbally. Unfortunately, very little is known about the extracellular field, as I have mentioned. In particular, it's really conjecture to assume that the location and the distance from the cell bodies follows similar patterns than what we know from the rodent literature. Human cell types are different in many respects. They are larger, they have different channels, and it's really necessary to repeat some of these classical experiments to confirm the validity of this type of experiment. So in particular, one would like to know what are the waveforms and the cell types, how does this correlate with anatomy and cell shape and dendritic tree structure, etc. So a long-term goal of my research is to combine this with en bloc resection and histology. So often it happens to be the case that the tissue that I recorded from where the microwires are located is actually later resected and removed from the brain. And because these wires stay in the brain for up to 3 weeks, I would assume that there is an immunological reaction that one could pick up to then later locate these wires. Now with this, I would like to thank the many people who have been instrumental in getting this work done. And I have highlighted a few people here, the students who most directly contributed to the experiments that I have shown today are these 3 people. Then I would in particular like to thank Adam Mamluk, who is my collaborator, neurosurgeon who performed all the surgeries, my collaborator Ralph Adolphs at Caltech, and my former advisor Erin Schumann for jump-starting this entire project when I was in graduate school. Thank you.